Hello again, Biology 3 students. We are moving into Chapter 4, which is cell structure. We are going to focus almost entirely on eukaryotic cell structure because that is the focus for much of the rest of the course. When we're talking about metabolism, or we're talking about genetics, we're talking about molecular biology, we're going to be looking primarily at those features in eukaryotic cells. We have images of three different types of cells here. In A, it is sinus cells, so these are eukaryotic, and it says it is a light microscope. The size bar tells us that that is 10 micrometers or 10 microns long. And so these cells look to be a little over 10 microns, and the dark purple circles are the nuclei. In part B, we have plant cells, and these are onion cells. I would have guessed without the label that they are plant cells just because of the very rigid structure. These were stained with, looks like some sort of blue dye. We can see the nucleus in the cells. What we don't see that's present in most eukaryotic cells that we see on a slide, or eukaryotic plant cells that we see on a slide, is we don't see any chloroplasts. These are plant cells, but they do not contain chloroplasts because they are onion cells, and that is a root structure of the plant that is underground, so there's no point in them having chloroplasts. But they are eukaryotic, they do have a nucleus, and the size bar looks to be about the same length, but that length represents 5 times as much distance. So these onion root cells are significantly larger than the nasal sinus cells. And in image C, we do have prokaryotic cells. Let's see. Vibrio tasmaniensis. This is why it's to your benefit that I can't hear you pronounce them. I still struggle. So it is a scanning electron microscope, much higher magnification, because once again, this bar is about the same length. But when this bar represented 500 microns, with the prokaryotic cell, that same length represents 500 nanometers. So that is 0.5 microns. So the bacterial cells, the prokaryotes, are extremely tiny relative to either of the eukaryotes. The microscope that we had the virtual exercise with was a compound light microscope. It is called a compound microscope because we have both the eyepiece that we look through and we have the objective lenses, the lenses that are close to the object on the stage. And we can typically look at things with a magnification between about 40-fold to 400-fold when looking at eukaryotic cells. The light microscopes that we have in lab and even that virtual microscope had up to a 1,000-fold magnification because the eyepiece has a 10-fold magnification and the objective lenses range from 4-fold to 10-fold to 40-fold and sometimes to 100-fold. So the maximum magnification we can get with a light microscope is around 1,000-fold. On the right-hand side, this is an electron microscope. And so the stage is not this whole table. This whole table is necessary to house all of the electronics needed to run the microscope. And this is the tower through which the electrons are passed. So it is called an electron microscope because rather than a beam of light passing through a specimen, a beam of electrons passes through a specimen. Because of that, we can get a much higher magnification of around 10,000-fold and some up to 100,000-fold and have a much higher resolution. And we didn't talk much about resolution in our virtual microscope lab, but resolution means the ability to see two items as distinct. How close together can they be, and we can still see them as two separate things. So the higher the resolution, the closer two items can be, and we would still be able to see them as two separate things. A light microscope has a resolution of about 200 nanometers, which is 0.2 micrometers. Electron microscopes have a resolution of 50 picometers. So a nanometer is 1 billionth of a meter, 10 to the minus 9. And a picometer is 10 to the third fold smaller than that, so 10 to the minus 12. So extremely high resolution images. Salmonella on the right are these tiny purple dots if we look at them through a light microscope. And this was probably a magnification close to 1,000-fold. The Salmonella are really, really tiny. They are only about 1 micron across. And we see here that our bar represents 25 microns, and each little dot, take 25 of them to fill up that space bar. So the tiny individual purple dots, those are the Salmonella bacteria. Those same bacteria, when viewed under a scanning electron microscope, look very huge. But our size bar tells us that they are a little over 1 micron across. Those are the pink Salmonella. These threads coming away are pili and or flagella. The flagella help the bacteria move, and the pili, that's spelled P-I-L-I, pili, help the bacteria to stick to their host cells. So the green ruffled cells are the villi in a human intestinal cell. We saw with our virtual microscope a cheek cell smear, human cheek cells that were stained with methylene blue, and that's what would appear like on the right. These are still epithelial cells, the type of cells that line our body cavities, so like our respiratory system, our digestive system, and our reproductive system are all lined with epithelial cells. These are from the uterine cervix, and they're taken from a Pap smear. It's called a Pap smear because this is done to collect the epithelial cells on the cervix, and those cells are turned over or they fall off at a regular basis, just like all of our epithelial cells. Right? We slough off the cells inside of our mouth, inside of our respiratory tract, and inside the reproductive tract. So normal epithelial cells look like this. They're kind of folded up on top of each other, but we can see individual cells, and each cell has its own nucleus. On the right-hand side, these are abnormal uterine cervix cells. This cell here is just really big. The nucleus is enlarged. These two cells each have 2 nuclei, and they are not actually in the process of dividing. A cell that is dividing and had 2 nuclei in it would look very different from this. So the Pap smear is looking for cells that are infected with human papillomavirus, HPV. That's why it's called a Pap smear, looking for cells that have been damaged by papillomavirus. So the HPV causes cell irregularities. It can disrupt the way these cells reproduce and can cause cervical cancer. This is a generic prokaryotic cell. Prokaryotic means before nucleus. So these cells have DNA. That DNA is a single circular chromosome, and the region where that DNA is sitting is called the nucleoid. So anywhere that DNA is, that is the nucleoid. There is no separate membrane around that DNA, so it is not a nucleus. That is only in eukaryotic cells. Prokaryotic cells, the pro meaning before and the karyo meaning nucleus, before the nucleus. They do not have any internal membranes. The only cell membrane they have is that cell plasma membrane. Outside of that, typically, prokaryotic cells have a cell wall. They might have a capsule, which is a gooey, slimy carbohydrate layer that is outside the wall. They may have pili, those projections that stick off and help it to stick to other cells, including host cells. They may have a flagellum. They may have more than one flagellum. More than one flagellum, you have many flagella, and that is used for movement. They do have ribosomes that are used in the processing and reading of RNA to make proteins. And that is your generic prokaryotic cell. Not all of them have pili, not all of them have flagella, not all of them have a capsule, but they do all have chromosomal DNA. What's not shown here is that they do also all have RNA. They all have ribosomes. Almost all of them have a cell wall, and when they do, it is made of a substance called peptidoglycan. So peptid tells you that there are peptides, that is amino acids linked together, and the glycan tells you that carbohydrate is present. So peptidoglycan is a sticky substance made of both peptides and sugars that form a prokaryotic cell wall. This image shows us some relative sizes, and it's a good thing that there is the size bar underneath it, because the images are certainly not drawn to scale, because an ostrich egg doesn't come up to the thigh of an adult female. But ostrich eggs are enormous. They are a single cell. All eggs are a single cell. So chicken eggs, ostrich eggs, pigeon eggs, reptile eggs. Frog eggs are about 1 millimeter, indicating these are things that we could see with the naked eye. Obviously, a human and chicken eggs. Frog eggs look like little gelatinous bubbles that collect along the edges of freshwater resources, some ponds, even some rivers. We might be able to see a human egg, but it would look like a little pinprick unless we had a microscope. The light microscope, we can see things that are 1 millimeter, but they would more than take up our entire field of view. Animal cells, we've seen the cheek cells through our virtual microscope. We saw the onion root tip through our virtual microscope. And then you did see a micrograph of the Elodia, the green plant that you had to do the annotation to show where the cell wall and the chloroplasts were. We can even see things as small as mitochondria. That's a specific type of organelle, a small structure within eukaryotic cells that has its own membrane. It is about the same size as bacteria at the 1 micron level. Getting down to the real limit, flu virus is one of the bigger viruses that exist, but it is just below the capacity of a light microscope to see it. We would need to get that greater magnification of an electron microscope to be able to see that. A virus is not a cell. A virus is only nucleic acid, so either DNA or RNA, but never both of those, and then a protein coat called a capsid. And some viruses additionally have an envelope, and that envelope is the previous host cell's membrane. So it's a phospholipid bilayer. We'll see that structure later in the lecture. Even some molecules can be seen with very powerful electron microscopes, and the most high resolution, high magnification microscopes, there are some of the larger atoms that can even be seen. Much later in the semester, we are going to do a DNA extraction. We'll watch one virtually and then have the grocery list, literally a grocery list for some items that you will need to extract DNA. We can do it either from wheat germ, which are plants, or strawberries. Either way, we grind up the plant matter and can separate out the DNA from the rest of the cell particles. Even if we had a light microscope to work with, you would not be able to see the molecules that compose that DNA that we extract. We will just see the gooiness of the DNA that's been pulled out of the cells. These images are to show us that if we could have either a perfect sphere or a perfect cube to represent a cell, that the surface area to volume ratio is really what determines how big a cell can get. And so we see that if we look at a cube, and we use a cube because that is mathematically the easiest surface area and volume to calculate. Because the surface area of a cube is the surface area of one side, which is the height times the width. So that'd be 1 times 1. And there are 6 sides on a cube. So that is why we'd have 6 millimeters squared. 1 millimeter times 1 millimeter is 1 millimeter squared. We multiply that by the 6 sides, we get the 6 millimeters squared. So that's the surface area. Volume of a cube is calculated by the length times the width times the height. Since this is a perfect cube, it'd be 1 times 1 times 1, which is still 1, but we have millimeters times millimeters times millimeters, and that would be millimeters cubed. So we'd have a volume of 1 millimeter cubed, surface area of 6 millimeters squared. So the surface area to volume ratio is 6 millimeters squared to 1 millimeter cubed. We represent that as just 6 to 1. If we double the length of every side, then our surface area is 2 times 2, which is 4 for one side. 4 times the 6 sides is 24. So our surface area is 24 millimeters squared. The volume would be 2 times 2 times 2, which is 8. And we cube the units because millimeters times millimeters times millimeters. So 8 millimeters cubed. So our surface area, which is 24, to volume, which is 8, if we have 24 to 8, we can reduce that and simplify it to 3 to 1. So if we double the length of one side, we have dramatically decreased our surface area to volume ratio because we increased our surface area from 6 to 24. That's a 4-fold increase, but our volume went from 1 to 8. It's an 8-fold increase. And if we keep doubling the size, eventually we get to where just the number that is in front of our units for the volume becomes larger than our number that represents the surface area. And then our surface area to volume ratio will be less than 1. That's important because reactions happen on the surface of cells. We have to be able to get materials into and out of the cell by osmosis and diffusion. And if we don't have enough surface area for the amount of fluid, then those reactions can't happen as quickly as they need to. I will not read every single little component on here, but we do have an animal cell and we have a plant cell. Now, this doesn't mean that every single animal cell will have every single one of these features, nor will every single plant cell look exactly like this one, but these are typical examples of that. The main thing, without even reading our labels, we can see that the animal cell doesn't have a definitive shape, whereas the plant cell is definitely square. They both have a nucleus. They both have these little jelly beans that are the mitochondria. But the plant cell has this big space in it that's called the central vacuole. And that is a way to control the water content in the cell and to maintain the pressure against the cell wall to keep this plant cell filled. The animal cell and the plant cell both have these little hair-like things inside them that are the cytoskeleton. Cyto means cell, so it is the cell skeleton. And we'll cover in the next slides the various organelles. So an organelle is like a small organ. It is a structure within a eukaryotic cell that has its own membrane and carries out a specific function. Having its own membrane means every time you have an organelle, more membrane means more surface area. And that is a way, without changing the size of the cell itself, we can increase its surface area by having organelles. We can also increase its surface area if the membrane was curvy. At the beginning of the lecture, we saw the salmonella cells in a human intestine, and the surface of that human intestinal cell was very wavy. That's another way to create greater surface area without increasing the volume that is inside the cell. So eukaryotic, EU means true or real, so it has a true or real karyonucleus. So the plasma membrane, which is also called the cell membrane, which is also called the cytoplasmic membrane, is a phospholipid. We saw that in our macromolecule chapters, the phospholipids, and it is a bilayer. All membranes have two layers of these phospholipids in it. And the polar heads face either the exterior or the interior of the cell, and the fatty acid tails face themselves in the interior of that membrane. We have integral membrane proteins, meaning they integrate all the way across both sides of that bilayer. We also have peripheral proteins. This one's stuck to the inside of the cell membrane. There are some that are stuck to the outside, but they don't cross that fatty acid bilayer. The thread-like structures shown here are the pieces of that cytoskeleton. So there is a particular component of the cytoskeleton that we will discuss later that forms the mesh that's directly under the plasma membrane that supports those phospholipid bilayers and gives it some more structure. We can have protein channels that are these integral membrane proteins, and that means they form a passageway across the membrane. We're going to talk about different kinds of transport, but these protein channels are what allows that. This little four-ring structure here is our cholesterol, because it is showing cholesterol, and it's a eukaryotic cell. We know that this is an animal cell. Only animal cells have cholesterol in it. So some cholesterol is embedded with that one single hydroxyl functional group embedded in the phosphate that's facing the interior of the cell. Other cholesterol molecules have their single hydroxyl in with the phosphate heads that are facing to the exterior of the cell. Other markers that can be on eukaryotic cell membrane surfaces, we can have glycolipids. So glyco tells you it is a sugar, and lipid, that is a lipid. So it's a lipid with carbohydrate. If we have a glycoprotein, it's a protein with a sugar attached. The microvilli is what we talked about just now regarding how a cell can make its membrane wavy or curvy to increase its surface area. So microvilli, small villi, little hair-like projections. That's an electron micrograph, a nice little cartoon. We can see that this membrane has a lot of surface area to it, and that is the side that's facing the inside of the small intestine. And so that is how we can get nutrient and waste exchange, by having a lot of surface area there. Now we'll get into the individual organelles and their functions. We're seeing the nucleus, and we are seeing the endoplasmic reticulum. It happens to be the rough endoplasmic reticulum because it has these little dots on it. Each of those dots represents a ribosome. The nucleus actually has two separate membranes. It has an inner membrane and an outer membrane. And we have pores where there is a gap in that outer membrane to allow the flow of molecules into and out of the nucleus. We have the nucleolus, which is a region inside the nucleus, and that is where ribosomes are made. Chromatin is how we describe the DNA, the chromosomes, when they are very loose and diffuse and the cell is not replicating. So that is the DNA inside the nucleus. The nucleoplasm is the fluid that is inside the nucleus. And the outer membrane of the nucleus is continuous with the membrane of the rough endoplasmic reticulum. That accomplishes two things. It keeps the rough endoplasmic reticulum attached to the nucleus, and that is important because in the nucleus, where we have our DNA, is also where the RNA is made. The RNA has to exit the nucleus to reach the ribosomes that will latch on to the RNA and translate that message that it carries so that the cell can build those proteins. There are also ribosomes that are free in the cytoplasm, but the RNA still has to exit the nucleus to get to those ribosomes. This is showing in a very cartoon manner how our eukaryotic DNA is organized. If you were to look at a dividing cell, the chromosomes do look kind of like X's, but that is highly condensed DNA. If we were to take our individual DNA molecules in our cells, get them all loose, stretch them out as much as we could, and lay them end to end, they'd be about 10 centimeters long. That clearly does not fit inside of our cells that are around 10 to 100 microns long. So the DNA, which is represented by this purple strand, is wound around histones, and histones are proteins. They're actually little 8 packs of proteins, and the DNA makes a specific double rotation around each of those little 8 packs of proteins, and that causes the DNA to not just be its coiled stepladder, but to coil and twist down on itself until it gets highly compacted. Why would a cell want to do that? Well, in our previous slide, we saw that the chromatin is just kind of spread out everywhere. And that works great while the cell is doing its normal functions. But if the cell needs to divide, we really need to organize that DNA and compact it so that it can be duplicated normally. And so when those duplications are made, they split and we get an absolute equal distribution of the DNA to both of the resulting cells. On the right side, we see actual chromosomes that have been stained so that we can see the pairs of chromosomes. Eukaryotic cells are almost all diploid, D-I-P-L-O-I-D, diploid. Di means two, and ploid refers to the number of sets of chromosomes. So each of us humans has received two sets of chromosomes. One set is entirely from our maternal parent, and the other set is directly from our paternal parent, regardless of the biological chromosomal sex. Half our chromosomes came from a paternal parent and half came from a maternal parent. And so these two big red chromosomes, we don't know which one came from the mom and which one came from the dad, but one's from mom and one's from dad. So in each of these pairs, one came from one parent and one from the other. Our chromosomes truly are different lengths. So these two have the exact same length. They carry information for the same characteristics, but they may carry variants of those characteristics. So first parent may have given the gene for curly hair and the other parent for straight. Curly's dominant. I got curly hair. Both parents may have given information for light-colored eyes. Both parents may have given information for dark-colored eyes. We will talk a lot about that in our genetics chapters, but we are looking at actual chromosomes from an individual. Those ribosomes that we saw in both the prokaryotic cell and the eukaryotic cell are made of two different subunits. Each subunit is made out of ribosomal RNA and protein. Eukaryotic ribosomes are much bigger than prokaryotic ribosomes. They contain a different set of ribosomal RNAs and proteins, but their overall shape is the same and their functions are identical. When we talk about how we're going to make the proteins, how is this ribosome going to sandwich around the messenger RNA and read it to make protein? Those processes are almost identical between the two cell types. So we see our big subunit and small subunit forming a sandwich on the messenger RNA. We have a transfer RNA that is holding on to our chain of amino acids during protein synthesis. This electron micrograph is showing a mitochondrion. There's our space bar. So a mitochondrion is about 1 micron across. The matrix is the fluid inside a mitochondrion. The cristae are where the membrane folds, and there are two membranes on each mitochondrion. The outer one, which is smooth, and the inner one, which is curvy. And those curves in the mitochondrial membrane also provide greater surface area for reactions. In our metabolism chapter, we will see the types of reactions that occur in the matrix and between the inner and outer membranes on the mitochondrion. Centrosomes, this whole image, are composed of two centrioles that are at right angles to each other. They're skewed to each other, and they are made out of microtubules. Microtubules are one of the components of our cytoskeleton. They are made out of alternating subunits of tubulin, and that creates the microtubule. It's a small tube, literally a hollow tube. And there are these triplets that are in between these other sort of spiked structures that form the centriole. They are also called the microtubule organizing center, where we have the production of additional microtubules when we need them to come out as these long, stringy structures that stick to chromosomes and push them to the middle of the cell to organize them during cell division, and then pull them to the opposite ends of the cell as cell division wraps up. Back to our macromolecules. This is cellulose. And you might think back, look back at your notes from chapter 3 with the carbohydrates. How would you know this is cellulose? Well, the six-sided ringed structure with a bunch of H's and OH's would indicate it's a carbohydrate. It's a polysaccharide because we have multiple subunits strung together. And we would know it's cellulose because the oxygen in the ring for this sugar is pointing downwards, and the oxygen in the ring for that sugar is upwards. Our glycosidic bond faces downwards here and upwards, so each of our subunits is upside down relative to its neighbor. So beta glucose links by the beta 1,4 linkages to make cellulose. The Endosymbiont Theory is part of our evolutionary concept. Endo means inside, and symbiont or symbiosis means living together. So we're talking about the theory of two things living together. Now that could describe a lot of things, but this is very specific to both the mitochondria and the chloroplasts. This is just showing a chloroplast. It has a smooth outer and a smooth inner membrane, and then it has a third membrane. The nucleus has two membranes. They're both fairly smooth. The mitochondrion has two membranes. The outer one is smooth and the inner one is wavy. The chloroplast has three: outer smooth, inner smooth, and then these little bitty ones inside are called the thylakoid. And if we have a stack of thylakoids, that is called a granum. The fluid inside of a chloroplast is called the stroma. So the mitochondrial chloroplasts are part of our endosymbiont theory. The concept that those two organelles used to be free-living prokaryotes. They are now no longer free-living, but they are integral parts of eukaryotic cells. We think they used to be free-living. We have ample evidence that they used to be free-living because they have their own DNA. And it is structured like prokaryotic DNA. It is a single circular chromosome. They have their own ribosomes. And those ribosomes are structured exactly like the RNA and proteins in prokaryotic cells. They are the same size as prokaryotic cells, and they divide independently of their host cells. So in us, if we're working out really hard, we're putting a lot of energy strain on our muscle cells. Those muscle cells will have mitochondria in them that are replicating at a faster pace than other cells, our livers, that may not be doing as much at the time that we're trying to run a half marathon. In plants, if they have a higher energy demand for photosynthesis, the structures that are doing more photosynthesis, that are closer to the outer edge of the plant, receiving more sunlight, they will have higher reproduction of their chloroplasts than the cells in the leaves that are not as directly impacted by the sun. This is another view of the interconnectivity of structures in eukaryotic cells. There's our nucleus the outer membrane of the nucleus is continuous with the rough endoplasmic reticulum. And then we have this additional structure shown here called the Golgi Apparatus, because Dr. Golgi figured it out. When you look at an electron micrograph, I would be hard-pressed to be able to look at it and tell you which one is which, except that the rough endoplasmic reticulum would be right next to the nucleus. The Golgi is, however, instead of wavy, continuous membrane, that's what the reticulum part of that word means. To be reticulated means to wave back and forth. The Golgi is separate flat sacs. They are not actually connected like our endoplasmic reticulum sacs are. And it's smooth. There are no ribosomes on it. It has a cis face, C-I-S, and it has a trans face. When we looked at our lipids, a cis fat had the hydrogens on the same side of the double bond, either both pointing upward, both pointing downward. So cis means on the same side. So this is on the same side as the rough endoplasmic reticulum. Trans, in our fats, meant one hydrogen was on the top and one hydrogen on the bottom. The hydrogens were across from each other, away from each other. And the trans face of the Golgi is away from the endoplasmic reticulum. So where the proteins are made by these ribosomes on the rough ER, we saw in chapter 3 that proteins have 4 levels of structure, or at least up to 4 levels of structure. They all have primary, secondary, and tertiary, and some even have quaternary. So we have a little protein coming off of the endoplasmic reticulum. It'll be pinched off in a piece of the membrane and fuse with the cis face of the Golgi, and it will get processed, finish its secondary, tertiary, and possibly quaternary structures. If it's a glycoprotein, it may have carbohydrates added to it. If it's a lipoprotein, it might get lipids added to it, and then it will be released from the trans face of the Golgi, again in a pinched off piece of the membrane that can fuse to the cell membrane and release that protein, or that membrane encircled protein can be transported elsewhere in the cell to have its final function. This electron micrograph is showing the nucleus, the nucleolus, where the ribosomes are made, this nuclear pore where we have a little breach in here, and then our rough endoplasmic reticulum that is connected to that outer nuclear membrane, and there happens to just be a mitochondrion that's sitting on top of the rough ER. There's the Golgi. Again, if I just showed me this without a label, it's like it's probably the Golgi because it looks kind of flat. I don't see any dots on it, and I do see these blobs right next to it that are either those pinched off pieces of the membrane from the rough ER fusing on the cis face, or if the nucleus is down here, it's the trans face and it's pinched off blobs being transported. This is an image of phagocytosis. So Phago means eat, and cytosis is a cell process. So it's the cell process of eating. Now our cells, of course, don't literally eat. They don't have a mouth that gobbles up and chews. They don't have a digestive system. But they do engulf and they do digest. So this is an image of a lysosome. If you see that S-O-M-E means sort of structure or bag of, and lyso means lysis or to break, like hydrolysis. So a lysosome is an organelle that has digestive enzymes. So it's a bag of digestive enzymes. In this case, it has engulfed a food particle because it has these pseudopods, fake feet, that literally wrap around a food particle. And when it does that, it brings it in with a piece of membrane wrapped around it. The lysosome, with its digestive enzymes, fuses with this food particle inside of a vacuole. That is when the enzymes become active and digest that food. We can use those molecules for cell function, and any waste can be eliminated. So this whole blue blob with the pseudopods is a macrophage. It is a type of white blood cell that is a big eater, macrophage. So in this case, the food particle is a bacterium. There's plenty of nutrition for a phagocyte, a macrophage, to eat. So even though that could cause disease, if our white blood cell macrophage engulfs that bacteria and chops it up, then it is inactivated and the cell gets nutrients and the waste can be excreted. These are the images of our cytoskeleton. We have microfilaments, those ones that we saw supporting right underneath the plasma membrane in a previous slide. The microtubules, this little cluster in the middle would be where the centrosome is, because the microtubules are made there and branch out. And then intermediate filaments are kind of all over the cell. They provide structure for the whole cell. They help hold organelles in place so that they can do their functions. This is a microfilament. It is made of the protein actin. And so this is all actin, and it's two strands of actin that wind around each other. And that's why it's called a microfilament. It is the smallest filament of our cytoskeleton. Intermediate filaments, because in terms of thickness, they are in between the microfilaments and the microtubules. But there are several intertwined strands, and it just says fibrous proteins because they are specific to the cell type where they're found. So the intermediate filaments in our skin cells are different than the intermediate filaments in our lung cells or our liver cells or our stomach cells. And it's a way to track the original cell source for tumors. So if I were to come down with skin cancer, if I had a melanoma, and they would take a biopsy, a small piece of my tissue, one of the things they would do, they would look at the DNA to see if it's been altered, but they'll also look at the intermediate filaments. And if the cells in that tumor on my skin have the intermediate filaments that are specific to skin, the bad news is I have cancer. The good news is that it originated in my skin and is staying in my skin. However, if they were to look at that skin tumor and find intermediate filaments that are only found in lung cells, then double bad news. I have a cancerous tumor that has spread from a different site. So that is one way that oncologists, cancer doctors, can determine if a cancer has spread. The microtubules are hollow. We saw them in the centrosome, forming those two independent centrioles. The dimer is two subunits. So a monomer is a single unit. A dimer is 2 units. And so there are 13 dimers of alpha tubulin and beta tubulin. So they alternate alpha, beta, alpha, beta, alpha, beta. There's 13, and then that keeps forming a spiral to make a hollow tube. These are flagella that are found in eukaryotic cells. The flagellum on a prokaryotic cell is made of something completely different, but we can see, this is a cross section kind of looking down the microtubules of the flagella. This says animal cell. My image is covering up the A. It's showing the extracellular matrix, so the outside the cell matrix of proteins and carbohydrates. There's our lipid bilayer. These are the microfilaments of the cytoskeleton. We've got a type of transmembrane protein called integrin because it integrates through both sides. We have fibronectin and we have collagen as two structures that are forming this complex outside of our cells. This proteoglycan, proteo tells you there's protein, glycan tells you there's carbohydrates, and there's a polysaccharide. All of this is providing protection to our animal cell membrane and cushioning to our animal cell membrane. Remember, we don't have cell walls. And so if we want to protect our cell membranes, we have an extracellular matrix that protects our cells. In plants, the connection between two cells is called a plasmodesma. If we have more than one plasmodesma, we have plasmodesmata. So the dark green represents the cell wall. This inner green here is the plasma membrane, the cell membrane. And then this yellow is the cytoplasm. And this gap in the cell wall is what allows the fluid exchange of the cytoplasm, but that means it also allows for nutrient and water exchange between cells. Animal cells have many different kinds of connections. One type is tight junctions, so they're watertight, sticking our cells together very, very tightly so that we are forming a sort of a single sheet of types of cells together to form a tissue. Desmosomes, they describe them as a type of spot weld. So you have intermediate filaments of this cell, and we have intermediate filaments of this cell, and then we have these structures called cadherins. That stands for cell, C for cell, and the AD to adhere. So cadherin, these proteins that stick our two cells together. And gap junctions are kind of like an iris diaphragm on a microscope where you can move it back and forth to let in more or less light. On a actual physical camera, whether digital or film, where you rotate it to open up the lens to let in more light, rotate it to close it to let in less light. These are not for letting light in or out. They are for letting chemicals and fluids in or out. So they can be closed, and that would prevent any exchange between these two cells. Under other cell conditions, they will physically move and form a channel that would allow exchange between these two cells. That is our whirlwind tour of the eukaryotic cell. You have, as you always will, until 11:59 a.m. on Wednesday, September 9th. I have to look that up. It is the 9th. Wednesday, September 9th, to do your initial post, and then you must return before 11:59 p.m. and respond to 2 of your classmates. I will be getting that second laboratory graded. If I see there was a lot of difficulty with the data set, I will open those back up so that people get another chance at it. This week's lab is chromatography and will be less detailed and does not have any graphing associated with it. So I hope you had a happy and safe Labor Day weekend, and I will see you on the discussion board.